Hey everyone, we're going to continue chapter 4, moving into talking briefly about the quaternary structure of proteins, now that we're talking about 3D structure, and then more into protein folding and methods to detect proteins. So we left off talking about the types of structure that exist in a protein, how we classify them. We talked about primary structure, which is just the sequence of amino acids. We talked about secondary structure, which are hydrogen bond formations that lead to patches of areas called alpha helices or beta sheets. And then we talked about the tertiary structure of proteins, which is kind of the final three-dimensional fold of the protein, which can be further held together by elements like more hydrogen bonding, ionic bonds, disulfide bridges, hydrophobic interactions that can kind of hold and compact the protein. So these are all the types of the main structure that we see, again, classified by primary, secondary, tertiary for the fold of a peptide. Now, the last one we're going to discuss again is quaternary structure, which not all proteins have, but many do. And this is any type of interaction that is formed by the assembly of individually folded polypeptides. And then these subunits kind of cluster together or interact together to form a complex. One really good example of a large structure like this that has quaternary structure is hemoglobin. So hemoglobin, which is shown here in this example, is made up of these 4 different subunits. These are all individual polypeptides that have been folded. And then these 4 separate subunits interact together and work as a complex. And we call that whole complex hemoglobin. So again, the name for the whole protein complex is hemoglobin, even though it is made up of subunits. And you will see this a lot with a variety of proteins. When we talk about their subunits, sometimes they'll be classified by letter, chain A, chain B, chain C, etc., or the alpha, beta, etc. chains. We're just talking about the subunit, which is a separate polypeptide chain that is then interacting kind of in one complex. Now, I gave you an example here, which is utilizing, looking at hemoglobin, and hemoglobin is a very large protein, but it doesn't matter the size of the particular polypeptide. You can still have quaternary structure in small polypeptides. So remember, it's any time you have the interaction of 2 separately folded chains. And one really good example of this is insulin, which you guys are all looking at. Insulin has an A chain and a B chain, which we call how we distinguish the 2 chains. They're both very small. They're very, very, very short chains, only about 20 to 30 amino acids long. Compared to hemoglobin, you can see here, this is just the cartoon representation of it, but each of those units is maybe on the scale of 100 or hundreds of amino acids long. Insulin has 2 short peptides. They each have their own small amount of folding, their own 3-dimensional structure that they have folded into. And then those 2 separate chains are interacting to form the main peptide, the main complex that we call insulin. So it's 2 chains that are interacted, and you can see they're only held together by these cysteine bonds. So the 2 chains of insulin have disulfide bridges that hold them together to interact and form their quaternary structure. So again, don't think that just because something is a small peptide, it can't have quaternary structure. It's anytime you have the interaction of two separately folded polypeptides. So moving on to talk about folding and protein stability in general, now that we kind of have looked at the classification of its 3D structure. Remember that a protein's function, its ability to do its job, is reliant on this 3D structure we have just discussed. So any loss of structural integrity will accompany a loss of activity. And we call that denaturation. Anytime you have a modification or loss of that folding structural integrity that then affects the protein's ability to do its job, or again, its activity. We use the word activity to talk about the job that a protein does, essentially. So we call that denaturation. And proteins can be denatured through a variety of means. You can denature them in extreme temperature, so heat or cold. You can denature them by putting them into extreme pH environments of acid or base. You can denature them with organic solvents, so things like acetone. And you can also denature them with other agents. Chaotropic agents is what we call them, like urea and guanidinium hydrochloride. These agents are going to specifically affect the disulfide bonds. And the reason for that is because chaotropic agents are going to affect the oxidative environment or the reducing environment of the protein. And those disulfide bonds will be will break or form depending on how reduced or oxidative the environment is. So protein folding for a protein in general can happen through a variety of means. There's different processes that kind of stabilize and help to fold proteins. And you can see some of these processes here. So you can use things called chaperones to help with protein folding or with keeping a protein folded. And these can also, again, help remodel a protein if it becomes misfolded. If a protein becomes misfolded, it also has other pathways that the biological system will take to deal with it if it cannot be refolded. One thing that can happen is it will be sent to a protease and then essentially fragmented up, and those fragments will be used as amino acids for fuel or for other things. Proteins can also, unfortunately, sometimes aggregate once they are misfolded, and this can lead to things like fibrils, unintended fibrils, or unintended aggregates, or unintended oligomers. And there are a variety of diseases that are in fact associated with misfolded proteins that have then aggregated into these states. Now, if the aggregation occur too much, they will then cause autophagy in a cell, killing the cell, and then eventually, again, be broken down into peptide fragments. So here you can kind of see, this is kind of just like the way that the body deals with protein folding and keeping it at a homeostasis, essentially, which we call proteostasis. So how they're assembled, degraded, etc., according to their state of being folded or potentially misfolded. Protein denaturation can be measured, and we often measure it to kind of get an idea of the stability of proteins. And as noted, proteins can be denatured in a variety of environments. So here we're looking at the percent unfolded of two different proteins. This is ribonuclease A and apomyoglobin. Myoglobin is a protein associated with oxygen transfer in other organisms that are not using hemoglobin. And you can see that as temperature is going up, we're going from a percent unfolded of 0, so this is good, this is folded protein, to a percent of 100 unfolded as you go up. And each curve for each protein is different. So this is showing you the difference in stability of ribonuclease A versus apomyoglobin with changing temperature. Now, one thing you'll see here is a dot with this TM, right? This is called the melting temperature. The melting temperature is essentially where you see this curve kind of straighten out. It's the midway point. And this is the point in which we describe the protein as beginning to denature. So melting temperature will be written and monitored often for proteins as a way to track where it will denature within a temperature, essentially. The second graph here is showing another type of denaturation that's occurring with ribonuclease A. So here ribonuclease A is denatured using this GDHCl, which is guanidine hydrochloride. So guanidine hydrochloride is going to instead disrupt those disulfide bonds. So if a protein does not held together with disulfide bond or does not utilize disulfide bond, then adding something like guanidine, this type of agent, this chaotropic agent, will not affect it. But you can see that ribonuclease A is in fact affected by the addition of this guanidine hydrochloride, which gives you a clue that it has disulfide bridges in it. And you can see again, as you increase the molarity of this chaotropic agent, the particular protein will become more unfolded. So we learned a little bit based on these graphs about the stability of these proteins. For example, apomyoglobin is stable at higher temperatures than ribonuclease A. And we also know now that ribonuclease A must have folding that is held by disulfide bridges because it is sensitive to the addition of a chaotropic agent. Ribonuclease, the example we were giving before, this particular protein is a small protein and it contains 8 cysteines and they are linked by disulfide bonds. So this data right here proves that there are disulfide bonds because its stability is affected by it. So again, other agents like urea or 2-mercaptoethanol, which is going to affect the reductive environment that the protein is in, will denature ribonuclease. If these are removed, ribonuclease can spontaneously refold, and it will correct the disulfide bonds that are reformed. This is not true for all proteins that have these disulfide bond types, but many proteins, when the oxidative or reductive environment is changed from one to the other, they can refold. And it will refold back into its native conformation. This is a very simple experiment. We were showing folding and refolding using this particular enzyme, ribonuclease. Essentially, again, addition of urea unfolds it, it becomes inactive, it becomes denatured, and then removal of urea, it will again refold properly, reforming those disulfide bridges and become catalytically active. Again, this experiment showed and proved using data like this, and as data with the graph here shows, that the main type of stability that this particular protein is held together with is disulfide bond linkages. Now, not all proteins can refold in this manner. And in fact, denaturing proteins with certain things like heat or pH will permanently denature them. One really good example of this is when you cook food. So eggs and egg whites and egg yolks are highly packed with protein. And when you heat that protein up to the point where you're cooking it, the protein becomes denatured. And when it becomes denatured, it changes color and it actually changes its texture. That's the protein unfolding. And that's when you see, for example, the egg whites go from this kind of clear state to a white color, or the egg yolks, again, going from that kind of more liquid yellow to a very solid state. Again, this is as the protein goes from its native folded state to an unfolded, denatured state. And many proteins, when you denature them, this type of structural difference will occur. And as you can see, you can't undenature eggs. If you cook them and you raise the temperature and you denature them, but then you let them cool, they will not spontaneously refold and go back to their original state. In fact, I'm very glad that they don't do that because it would be kind of gross. Now, when proteins fold, when they're biologically active in their native state, they fold in a particular pathway or sequence. These folding patterns of beta sheets and alpha helices don't just snap fold like that. They go through a sequence in which smaller areas are first folded, things like small units of alpha helices, one or two beta sheets. These then maybe interact with each other. And through these steps, they finalize their particular motif, folding motif of these alpha beta sheets that are then linked together. Now, I mentioned a little bit about a particular type of protein called a chaperone, and in relation to being able to refold these proteins. So within a biological system, when a protein potentially has some problem with misfolding, or in general, just to help it get to its properly folded state once it has been made, the body and biological systems will utilize things called chaperones. So they will prevent misfolding from happening. They help prevent aggregation, these fibrils that could potentially happen, or a bunch of unfolded protein that can then aggregate into these clusters, which we do not want because that will cause cells to die. So these chaperones will take kind of a partially folded or folded intermediate of a protein and then help it to stay folded into its native state. And it does this through the use of another protein complex and ATP, which it utilizes as energy. So you can see the chaperones work like this. Chaperone proteins tend to be two main subunit proteins. These proteins are huge, let me say. They're represented here in a cartoon version, but they're made of lots of polypeptide chains, which are interacting in some type of quaternary structure. And they have multiple subunits that then form this main complex. But looking at it in this cartoon form, we can think of this as one complex on the bottom and another complex on the top. And each of these complexes can take a protein and help to fold it into its native state if it is misfolded. But they do them interchangeably. So when the top portion is working and folding a protein, the bottom portion is regenerating, and then vice versa. So you can see here, we enter, if you insert a misfolded protein and utilize 7 ATP of energy, this is going to use up the ATP in the top subunit and release the native protein after being folded. At this point, the bottom subunit can then intake a protein to help with its folding. Here's an actual image. This is a image you can look at, for example, on the PDB of the structure of a chaperone protein, and it's a chaperone protein with other proteins in it that is helping to fold. So you can see, in fact, how large it is. This is kind of that top subunit here. It has this like cap portion and then the main body portion, which are made up of many other polypeptides that are folded and interacting together. And then the inside here is a protein, this GP23, which has just been newly folded and into its proper state. And you can see in the bottom, getting ready once this protein is released, is a unfolded or non-native state GP23. So it has entered the bottom portion of the chaperone and it is being ready to be folded. So as I noted, protein misfolding, this ability to denature or misfold, can occur in a number of human diseases. So one very, very good example of this is the beta amyloid protein, which is associated with Alzheimer's disease. Beta amyloid, as shown here, when it's in its native folding structure, is a globular protein. Remember what we talked about, fibrous versus globular protein. So you can see it has this kind of spherical nature. It's a mixture of beta and alpha chains. This protein can potentially misfold, and when it misfolds partially, the chains on its secondary structure have the potential to reorient and restructure themselves into an aggregation or fibril type structure. So they go from this native globular protein to potentially having this fibril nature as they interact with other beta amyloid structures that have been misfolded. So at this point, the unfolded structure can actually influence other natively folded beta amyloids to unfold and denature and then fold into this amyloid fibril structure. These structures will then further assemble and they become these very large, what we call amyloid fibrils. And the correctly folded helices and all of the original alpha helices that were present in the beta structure are actually lost and they form instead these large beta strands and what we call beta helices. So they're beta sheets that are wrapped around each other. This, of course, when this occurs, you get these large fibril structures. Think essentially if you had a structure like keratin in your hair, but now it is growing into this long, very strong fibril structure within a cell. That will, of course, cause autophagy, and this unfortunately tends to happen in things like neurons within the brain. So you're having loss of neurons through the formation of these amyloid fibers that are occurring. So again, just a look at how important protein folding is within biology in organisms.